I've been talking a lot about viewing things on the genome browser from the variation service job. So here's the way you do it. Up here is the browser icon. Let's click on that. And this goes to the genome that has been annotated in BV-BRC. You have to have the genome annotated in the resource. Not only because we look at individual SNPs as they are on the annotated genes, but it just provides all this other functionality like the browser, the circular view, which we'll do next, and just being able to dig into all the other information that's there. Like when you look at the feature view and it tells you what pathway or subsystem that gene is on. So here it is. If you look at this, these are all the contigs that are in this genome. And it chose to open on the 6th, which is fine. And it's showing you the entire contig sequence. You can see that red bar is an indication that you're seeing the whole thing. And some of it is just missing some annotation. So there are BAM files and there are VCF files. The VCF files are showing the individual SNPs. Let's take off the RefSeq annotation. I can take it off by clicking here. And so let's click on the first one. This is for the first sample, which was 96. And it's showing you where all those SNPs are called on that across that entire contig. And I can add in the second one. And look, a bunch of them are piling up here. This first one has a bunch of unique mutations here, and you can see that these are piling up and sort of a similar pattern. And then we can add in the third one here. So all of these are piling up in the same area of this genome. We don't know that it's a gene yet. So let's start zooming in. So I click on that. And it's bringing me into a particular region, and I want this. Now I can scroll over with my mouse and get more to an individual region. And I can go in a bit more, and then I can see individual genes. And let's go in one more. You can do a large zoom or a smaller zoom here. This is a smaller zoom. And look, all of these are in this gene. And when I mouse over it, what does it say? Dihydrofolate reductase. This enzyme is vitally important in folate synthesis. And if these isolates were grown in trimethoprim, which can kill the bacterium, and it's this enzyme that they're acting on. So that's where you're seeing the majority of changes are in that particular gene. And strong indication that it's under pressure here to respond. You either have a good mutation that will make you resistant if it's a bacterium, or you're going to die. So it's showing you the area that this is in. I want to zoom in here and put in the ref sequence. May take a bit to load. And some of them are at the beginning of the gene, and some of them are later on in the gene. I guess we're going to have to zoom in even more. Then you start seeing, let's go over where there's a lot of action. You can zoom in to where you can see the 6-frame translation of those. So pretty sweet, right? Pretty nice. Now, if you're interested in, I'm going to click this, I'll click these guys off, the other ones, because I want to show you if you click on the BAM file, you can see how deep the reads are in those particular areas. So it's got to be pretty zoomed in to get this particular view. And look, it's even showing you the location of those reads. And so you can see how deep that is. And compared to other regions and part of this genome, that's pretty good, right? And look at this. Here's another unique functionality. I can click on this thing. That's an individual read, and it's giving me the information on that particular read. So this is how you can see the information, see where the SNPs are, and see how deep the coverage is for them using BV-BRC. And so how sweet is that? I love the genome browser. I use it all the time. The main takeaways are if you want to read the BAM files, you've got to zoom in. If you do it too early, it'll say it's too much data and it can't do it. So in the next one, we're going to look at the circular viewer. I will see you there.